A chemical known as dopamine is released from an area of the brain called the substantia nigra.
This is how the brain works. In the center part of the brain is where the cells that produce serotonin and norepinephrine modify the rest of the brain. Just like a dimmer switch can control how light or dark a whole room is just from one small area, each cell has connections with 500,000 other cells, and you only have 250,000 in your whole brain. The signal comes down the nerve. It releases these chemicals, which then go across the space called the synapse. Once those chemicals fill up the receptors on the left side of the synapse, the signal is sent. That's how the nerve on the right influences the nerve on the left. Here's a higher magnification of the same thing. The signal comes down the nerve, it releases, goes across, reaches critical mass, now sucks back up into the yellow vacuum system. Now the body will recycle some of it, but the rest of it is destroyed and thrown away. And how much is destroyed and thrown away is dependent upon your family characteristics. So if you don't have enough of the chemical to fill up those receptors, then you will have the signs and symptoms of depression and anxiety. Some people have problems with too much rather than too little. This is what we think happens with people with schizophrenia, and this is the dopamine system. They're being so overstimulated with dopamine, they become very agitated, they hallucinate, they can't sleep, they hear voices. So what we use is medications called atypical antipsychotics, which are represented by these green balls, which temporarily sit and block those receptors from being overstimulated. Helps to calm them down, the voices go away, they can sleep much better if they'll take their medication. Now this is what happens with depression. Chemical is released, it did not reach critical mass, so it sucks back up into the yellow vacuum system, and a double dose is now released. Now there's enough to fill up those receptors, and boom, the signal is sent, and now it's sucked back up into the vacuum system again, waiting for the next signal. Now antidepressants are not addicting. All they are these white balls that simply plug up that vacuum system. So now the body can't bring it back in and throw it away. So it helps your body conserve what you've already made. So now the concentration in that gap increases. It's like having instant hot water. When you turn on the spigot, you have hot water immediately. You don't have to wait for all the cold water to be pushed out of the hot water pipes before you get a cup of coffee or tea. This is what's addicting. This is in the pleasure center of the brain, and these are the dopamine receptors. And these purple balls represent methamphetamine and cocaine, which now begin to hit and to smash and to destroy those receptors. And it's like taking a hammer and smashing it through a board of wood so that people can no longer experience normal pleasure and joy in life unless you have more and more cocaine. So if you understand that, you understand how the brain works.
Deep brain stimulation is a surgical technique. It's a procedure. What we do is we implant electrodes or wires in the brain in very specific areas, and we use those wires to deliver a very fine electrical current to those regions of the brain in order to reduce the symptoms and the signs of Parkinson's disease and other movement disorders. The electrodes in the brain are connected up to a battery which is implanted under the skin just below the collarbone in most cases. That battery allows us to deliver a continuous small electrical current to the brain in a very controlled fashion in order to reduce the symptoms and signs of Parkinson's disease or other movement disorders. Despite the fact that it's called deep brain stimulation, in most cases we're not actually stimulating the brain, we're shutting down areas of the brain. By delivering an electrical current which is a fairly high frequency current, the nerve cells in the area of the electrode stop working, and that allows us to fairly reliably and accurately shut down a small volume of brain around a particular part of the electrode. So even though we're delivering an electrical current, we're actually inhibiting the brain or shutting it down rather than stimulating it.
Well, the mysteries of the brain are unraveling at a record pace. Scientists are mapping and plotting coordinates in the brain to identify areas that control various body functions. Then surgeons target these coordinates when treating diseases such as depression or Parkinson's. Deep brain stimulation is a kind of pacemaker. Extremely fine probes are inserted into the proper area, and an electrical pulse is provided to the brain. These pulses stimulate the brain and provide a measure of control for patients that was simply not available prior to DBS.
Electronics in the body, they need to be made as small as possible. Most devices are powered by batteries which are very bulky. To eliminate them, inductively coupled coils have been used to wirelessly transfer power. However, the electromagnetic field decays quickly, requiring large receiving coils. Midfield wireless powering overcomes these problems. A flat structure interacts with tissue, inducing propagating waves that converge on a micro-device implanted deep in the body. A micro-implant consisting of a power harvesting coil, integrated circuits, electrodes, and fixation structures, all packed into a device the size of a grain of rice, can now be implanted and safely powered almost anywhere in the body.
Wow. The wand works by producing a powerful magnetic pulse.
So it doesn't look like a lot, but the magnetic field that it generates is about the strength of an MRI machine, of a very strong MRI machine.
Since electricity and magnetism are really just two forms of the same thing, a magnet can affect the electrical signals in your brain. Now this is your brain. It's basically an electric web of billions of neurons wired together. When a strong magnetic pulse hits these neurons, it alters their electric current. The process is called transcranial magnetic stimulation, or TMS.
Electricity is the currency of the brain. All thoughts, all beliefs, all actions are just electrical impulses. And so TMS, we're actually able to get in there and influence the currency of the brain focally and non-invasively.
The stronger the magnetic pulse, the deeper into the brain it goes. And by adjusting the pattern of the pulse, you can change the way that part of the brain functions.
We can turn a part of the brain up or down or temporarily turn it off. It doesn't take a genius to see that it should be a pretty fertile way to begin to understand how the brain works.
One mystery George wanted to decipher was how the brain
algae called Chlamydomonas. Chlamydomonas needs light for photosynthesis, and it swims toward light with the help of a small organelle called an eye spot. The eye spot contains a light-sensitive protein called channel rhodopsin, which responds to blue light by moving positively charged ions across the eye spot membrane, causing the voltage of the eye spot to change, a bit like a battery being charged by a solar cell. But neurons are also electrical devices, and their voltage would also change if channel rhodopsin could be inserted into their membranes and then illuminated. So, researchers took a piece of DNA that encodes for the channel rhodopsin protein. Then, they put it into neurons, using a safe and effective method for delivering a gene, the same kind being used in human gene therapy trials. The neurons expressed the DNA, manufacturing copies of the channel rhodopsin protein and installing them in their membranes. As predicted, the researchers were now able to activate these neurons using light, without affecting any of the neighboring neurons. This technique, called optogenetics, allows researchers to drive the activity of specific neuron types to see how they affect brain activity and behavior. For example, researchers can deliver the piece of DNA that encodes for channel rhodopsin into a set of basket cells, shown here in green. Then pulses of blue light activate only the basket cells, while the other cells, for example the neighboring pyramidal cells, are not affected. So, if a specific brain disorder is associated with dysfunctional basket cells, it would be possible to augment the activity of these cells and possibly repair brain function. And these tools don't just activate neurons, they can silence them as well. Archirhodopsin and halorhodopsin, for example, are proteins that are sensitive to orange light and produce the opposite effect to channel rhodopsin. When researchers deliver DNA that encodes for one of these molecules into a specific set of cells and shine orange light within the brain, they can turn off just the selected set of cells. By combining these tools, along with others now being developed, researchers will be able to control brain activity with a degree of precision that would have been unimaginable a few years ago. Optogenetics is currently being used in animal models to understand how normal circuits work, but it may also lead to completely new types of prosthetics for treating a wide range of human brain disorders.